Well, in each case, the Shine-Dalgarno sequence is approximately 10 base pairs or 10 bases away from the start codon. So by putting that thing in its proper context, the ribosome knows which AUG to use. You'll notice there's a GUG there. In very rare cases, GUG is used as a start codon. I don't know why they bother to put it on there because it's not a very important one, but it does happen sometimes. Okay. So let's go forward. We've just finished the process of initiation. Now I'd like to talk about elongation. And this, again, looks worse than it is. When we finished the last step, we had an assembled ribosome. We had a messenger RNA going through it. We had a transfer RNA, the initiator tRNA, in the P site holding on to a formulated methionine. Now we're going to elongate, meaning we're going to start adding amino acids to this growing polypeptide chain. Here's the A, there's the P, there's the E. Okay? What does all this blah, blah, blah tell us? Okay? It tells us, first of all, that we've got to get another tRNA in, and the tRNA's got to go into the A site. Does just any tRNA go in there? No. It's got to be the tRNA that is complementary to the codon that's in the A site. How do we ensure that the proper one goes in there? One of the ways we ensure, and this is true for us, it's also true for E. coli, which is what this is about. One of the ways that we ensure that that happens is there's a protein called EFTU. EFTU carries the tRNA to the A site. It carries the tRNA to the A site. It makes sure that the base pairs are properly formed. So, for example, if it's brought the wrong one and the base pairs aren't the right ones, EFTU will take it away and nothing will happen. On the other hand, if the proper one is left there, then EFTU will cleave GTP again. It will cleave GTP and it will exit. It'll get out of the way. Now you might wonder, why does it cleave GTP? What's it doing? Well, it turns out that the EFTU, when it brings that tRNA to the ribosome, it's covering up the bond that's between the amino acid and the tRNA. It's actually sitting on top of it. And to get it off of there requires energy. Why is it sitting on top of that bond? Because that bond is very, very unstable. It's very, very unstable. It's unstable in water. And that's what all this stuff is floating in. So EFTU is covering up the tRNA amino acid so that that bond never sees water. You might say, well, wait a minute. When it got made, wasn't it in water? Yes, it was. But EFTU had to go on to it almost immediately. And for that reason, EFTU has to be very, very abundant in the cell. The cell has to have plenty of it to be ready to glom on as soon as that bond is made. It turns out that in E. coli, EFTU is the most abundant protein found in E. coli. Yes, sir?
It covers up the bonds on the codon or the anti-codon?
It covers up the bonds between the tRNA and the amino acid.
So it doesn't, oh, okay.
Okay? So it has nothing. So the tRNA, of course, has an anti-codon, but that has nothing to do with EFTU. EFTU is covering the bond between the amino acid and the transfer RNA. Okay, so EFTU has to be abundant. In this case, we've got a proper base pair for the thing that's in the A site. Okay, so here's the one in the P site, here's the one in the A site. The rest of it is real simple. We're not going to worry about the rest of this blah blah over here. Okay, we're going to go down here and look at what happens in the next step. We're going to make a covalent bond between the amino acid that's on here and the amino acid that's right there. Notice we've now shifted. This methionine is now attached to this amino acid, which I think is alanine. Okay? It doesn't have to be alanine. In this case, it just is. We've just made a peptide bond. We have just made a peptide bond. We have just elongated that amino acid chain. Notice now that this tRNA is holding on to the entire polypeptide chain. The one that was in the P site doesn't have anything on it anymore. It's going to get shoved out of the way, and it's going to go out the E site, which E stands for exit. Okay. How did that peptide bond get formed? That peptide bond got formed by something very, very interesting and cool, and it's something we didn't know about until about the 1990s. Okay? That peptide bond was formed by action of a catalyst that's actually the 23S ribosomal RNA. RNA can be a catalyst. It turns out that all the peptide bonds on the face of the Earth are formed by catalytic action of 23S ribosomal RNA in bacteria. In us, it's called 27, but we'll talk about that later. Okay? That is why one of the reasons that ribosomes have RNA in there. That catalytic action is necessary to make peptide bonds, and the RNA catalyzes that formation. Okay, we're almost done. We next are ready to move down one more codon. So the ribosome now has said, okay, we're done here. We're going to slide down one more codon. To slide down takes energy. The energy it takes is supplied by another G protein called EFG. Okay. By the way, these are all G. Don't worry about it. I'll explain that later. But another protein called EFG, and EFG uses energy from GTP to slide that ribosome down one more. So you see now it has moved one more to the right. What was in the P site was this empty tRNA. It has now gone to the E site, and it has exited. And we're right back where we started. We've got something in the P site, and we're ready to bring in another tRNA. So yes, this is an intricate process. It is a very intricate process. It's amazing. This is a little molecular machine that's making this. It's a little factory. Okay, so elongation goes on and on and on until a stop codon appears in the A site. Okay, so we'll just keep repeating this process over and over and over until a stop codon appears in the A site. When the stop codon appears in the A site, the ribosome stops. And the reason it stops is that there's no tRNA that will form a stable base pair with it. Okay? There is no tRNA that will form a stable base pair with the with the stop codon in the A site. Well, what happens then? What happens is that there are proteins called release factors that come and they bind to the A site. So they're going to facilitate the termination of translation. Release factors. And yes, there are several of them as well. The numbers won't matter for our purposes. How do the release factors work? In a very simple fashion. They bring a single molecule of water. And guess where they put it? Right at that bond between the polypeptide chain and the tRNA. They break that bond. The polypeptide chain is released. And everything falls apart. That is termination. Now, that's a little bit more detailed than you're going to get, I think, in most biology classes, and I understand that's a lot of information that's right there. So look through it. If you have questions, let me know. Come talk to me. I'll be happy to sketch it out for you or tell you more. Yes, sir? Can I repeat termination? Yes. So termination occurs when the ribosome, during the elongation phase, slides down and a stop codon appears in the A site. So when that stop codon appears in the A site, the ribosome stops because there's no tRNA that can come in and make base pairs with it. No tRNA exists that forms base pairs with the stop codons. That means then that it allows the release factors, which are these proteins, to come in and carry a molecule of water to break the bond between the polypeptide chain and the tRNA that's in the P site, because that's where that long polypeptide chain is now. That allows the polypeptide chain to be released, and then everything falls apart. Did you have a question?
So the 23S is supplied by a large volume.
23S is a part of the 50S subunit. That's correct. Yes, sir.
Are the release factors waiting for a pause in production?
The release factors are doing exactly that. They're waiting for their opportunity, waiting for a pause. That's right. They actually do form a sort of a base pair with the stop codon. So they have a structure that looks kind of like that. The most important thing is that they're carrying water. But yes, they do form a sort of a bond there. Yes? Yes. I'm not sure I understand the question. Yes. The one that starts in the P site is the initiator Trna. The initiator's tRNA starts in the P site. Every other one comes in through the A site. Okay?
And is there, you said the E for exit, is there any other way to remember
I just remember ape. Comes in A, goes to P, and exits E. Okay? Yes, Matt.
So does the EFTU protein, you're saying that's the one that blocks the, is it trying to prevent a premature termination?
It's trying to keep that amino acid on the transfer RNA. Because if water sees it, then the bond gets cleaved, and there's no amino acid on there. So that's why it's covering it.
And during termination, does it disappear? Or?
So it disappears once the proper base pairing is formed. So it's, remember I said it's leaving. So when it's leaving that amino acid attached to the transfer in the sort of belly of the ribosome. There's no water in there. Okay. So it's brought it to its destination. It's exited, and it's left it in a place where there's no water. Make sense? And so the only way water gets there is when the release factor brings the water in and now breaks that bond. It's a cool process. Okay, that's a lot of information. What do you say we sing a song? This is a song I wrote several years ago, and it sort of covers a little bit of each of these processes. And it's to the tune of YMCA. You guys know the song YMCA? Okay, I want to hear you, right? Yep. It's called BDNA. Phosphates are in nucleotides, I say. Phosphates cover bases inside, I say. Phosphates span the 5 and 3 primes. There's no need to be real mixed up bases. Carry info, you see, I say. Bases are all complementary, I say. Bases like A, T, G, and C, they have got to be all paired up. Bow, 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 bow. It's fun to play with some BDNA. It's got a boatload of GCTA. It's got everything a polymerase needs when you melt all the A's and the T's. It's fun to play with some BDNA. It's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's with A's. Proteins full of amino A's, I say. Proteins come from mRNAs, I say. Proteins require tRNAs. There is more you need to translate codons. Like our friend UAC, I say. Codons come in clusters of 3, I say. Codons have one base wobbly. Now you can go forth and translate. Bow, 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 bow. It's fun to play with some BDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s.